Hello and welcome to the first very awkward lecture online. So this is ironically the part of the semester where we switch from talking about bacterial pathogens to learning about viral pathogens. And I give this lecture using influenza as an example. We're all familiar with influenza, we've heard about it. And it's a really good one to go through viral structures and replications. And so just some quick stats for this current year, right? Flu's not being talked about much right now because of the coronavirus, but these are stats just for the United States, where we estimate there's been somewhere between 36 and 51 million influenza illnesses, approximately 500,000 hospitalizations, and somewhere in the range of 22,000 to 55,000 deaths from influenza in the United States. And again, that's just from October through the 7th of March. So it's, even though we're not talking about it much this year, it's still a really, really ongoing big deal in our country this year. So here would be our starter case study. There's been an outbreak of pneumonia. A local nursing home is experiencing an outbreak of bacterial pneumonia in its residents. In the previous 6 weeks, there had been a similar outbreak of a viral respiratory infection in the facility. So I'd ask you to pause the recording here and think to yourself, predict what might the original viral pathogen have been that infected the residents, and why do you suppose that initial viral infection predisposed residents to a secondary bacterial infection? So I encourage you to pause it here, think about those two questions for a minute, and then continue on. All right, so today, this is going to be kind of a longer one. But today we're going to learn the different morphologies of viruses. So what different shapes can they take? What different structures can they have? We're going to talk about the different types of genomes that viruses can have. This is, in fact, what makes viruses the most diverse group of organisms on the planet. We'll talk today about the lytic cycle of replication, and in a few weeks we'll talk about the latent cycle of replication. And how these two cycles cause differences in disease. I want you to identify any new terminology for understanding viruses that you don't already know, and to be able to explain the structure and replication of influenza viruses in particular. And then we're going to end this lecture by talking about the differences between genetic shift and genetic drift, and the role that they play in viral diseases, and in particular in influenza outbreaks and the emergence of new influenza strains. Okay, so viruses themselves are obligate intracellular pathogens. They're what we call acellular organisms, meaning they're not even a full cell. They are unable to generate their own ATP. They don't use any metabolic pathways. So they use host nucleotides, host amino acids. They don't build these things on their own. They just use what the host provides for them. And many also use host enzymes for replication. So all viruses that we have discovered use host ribosomes. None of them encode for their own ribosomes. Many of the viruses that have DNA genomes use host DNA polymerases. They almost all use host RNA polymerase II to make their messenger RNAs. Some of them even use the helicases from their host cells. They'll use the enzyme primase and a variety of other enzymes in order to replicate their genomes, make their proteins, and make more viruses. So they have to live inside a cell because they can't do all of these things on their own. Oh, look, I left this one part down. Sorry about that. When we classify viruses, we don't use all of the same levels of classification we use for our animals and plants. So all of the kingdom, phylum, class, order, family, genus, species, we don't use all of those for viruses. We really only use 4 levels and then sometimes a sub-level within that. So the first is order. So the name of the order ends in ales. So I'm going to tell you the classification for coronavirus. They belong to the order Nidovirales. So virales like viruses. Next down is family. And you'll see this one more often. Family ends in this suffix viridae. So our example here would be the coronaviridae. From there, we do go down to the genus level. This ends in virus. So for our coronaviruses, we have these for genera, the alpha coronaviruses, beta coronaviruses, gamma coronaviruses, and delta coronaviruses. And from there, we do actually break it down into individual species. So the viral species for the beta coronaviruses are labeled as such. So HCoV, that stands for human coronavirus, HKU1, human coronavirus OC43, SARS-CoV, which emerged in 2001, 2002, and then SARS-CoV-2, so SARS coronavirus 2, which is the current one we're dealing with. So this is the name of the virus, SARS-CoV-2, and when you hear COVID-19, that stands for coronavirus disease 19, that is referring to the disease symptoms that people experience when they are infected with the virus SARS-CoV-2. So these are two distinct terms, but they're currently being used interchangeably in the media. Right, so you guys know, you've heard before that viruses are super, super small. We can't see them with our regular microscopes. We have to use electron microscopes to see them. But I want you guys to appreciate and have an idea of just how tiny viruses are. So up here in our top left corner, we have a red blood cell. This oblong bacillus is E. coli. And all these little dots down here are viruses. So we know, you guys have looked at E. coli under a microscope, you know how small it is. The viruses are infinitely smaller than our bacteria. So if we zoom in on E. coli, this big blob here is smallpox virus. This is one of the largest animal viruses so far identified. This alien robot-looking thing is a bacteriophage. Bacteriophages also come in various shapes and sizes. This is the more famous one because it's frankly really cool looking. These other dots that we see here, still too small to make out shapes. Those are some other viruses. So if we zoom in now where our smallpox virus is nice and big, you can start to make out the structures of other viruses. So here we have tobacco mosaic virus. That was actually the first virus ever identified back in the late 1800s. Here's that alien robot bacteriophage T4. Here's another bacteriophage, really super tiny here. And this little guy is poliovirus, right? We're currently working on eradicating polio from the world. It is about 30 nanometers in size. These are ribosomes. They are 25 nanometers. So poliovirus in itself is not much bigger than a bacterial ribosome, but it causes pretty terrible disease. Whereas smallpox over here, which also causes really terrible disease, is a lot larger. So we can see a really great variety in shape and in size between our different kinds of viruses. Now viruses, they're very small. They're also structurally very simple as compared to our cells, right? They don't have mitochondria, they don't have nuclei. Viruses really all have two structures. The first is that they all have a capsid. This is a protein shell that protects the viral genome. So our capsid is depicted here in this sort of teal color, like this. This one that has definite planes, it looks like something that you learned how to draw in organic chemistry. This is what we call an icosahedral capsid. They can also have helical capsids. That's what this one is showing. Each of these little sort of beads on the string are the capsid proteins that surround the RNA. So the RNA is this sort of orangish line inside that nucleocapsid called complex there. And then we have a set of capsids that are called complex capsids. That means they're neither icosahedral nor helical, and they usually have some other shape, or they have some additional components to them. And I'll show you a couple of examples of those. So all viruses have a capsid, and that capsid protects their genome. Now this is where viruses get very different from cellular life, from bacteria, humans, animals, insects. Their genomes can be made of DNA, just like ours, or they can be made of RNA. And within these two larger groups, there are a bunch of subgroups. So they can be single-stranded DNA or RNA. They can be double-stranded DNA or RNA. Their genomes can be linear, like we see in all of these pictures here, where they have two separate ends, or they can be circular, like a bacterial chromosome is circular. They can be segmented, meaning that the virus may have more than one distinct pieces that makes up their genome, kind of analogous to chromosomes. Or they can be continuous, like all of these are, where all of their genetic information is contained on one nucleic acid. They can have a different sense, so they can be positive sense, meaning that they look like a messenger RNA. They can be antisense, meaning they look the opposite of a messenger RNA, or they can be ambisense, meaning that part of their genome looks like a messenger RNA and part looks like not a messenger RNA. So there's a huge deal of variety just among the genome types, the genome makeup of our viruses. So all viruses have a capsid that protects their genome. Some viruses have an envelope. So an envelope is the virus word for a membrane. So we can see here, this one does not have an envelope. We call it a naked virus or a non-enveloped virus, whereas these two have envelopes, okay? So that envelope is a lipid bilayer that comes from the host cell at some point during viral exit from the cell. So this membrane that this enveloped virus has here came from the cell. Same for this enveloped virus, even though their capsids are different. And we can see that within these envelopes, they have a number of proteins sticking up, these little green proteins. We call these spike proteins. These are going to be what the virus uses to attach to a host cell. And over here on our non-enveloped virus, if it has spikes like this, that's going to be what it uses to attach to the host cell too. So to give you some examples of what these look like and to put them kind of in context of viruses you may have heard of, here we have our helical capsids. So this is in this case, a very rigid capsid for tobacco mosaic virus, and its genome is threaded through this. So I kind of like to describe these as if you took a pencil and you wrapped a pipe cleaner around it very tightly until the whole pencil was covered with the pipe cleaner. The pencil would be your genome, the pipe cleaner would be the helical capsid. So these are really rigid, they don't bend a lot. Over here we have a picture of Marburg virus, which is closely related to Ebola. This is also helical, and you can see they do make straight lines like this, but they're flexible in this case, and they can actually bend and make loops or shepherd's crook kind of look, which is what we think of when we think of Ebola. Down here we have adenovirus. This is an icosahedral, also called polyhedral capsid. Adenovirus, we've all had adenovirus at some point. They cause the common cold, they cause viral pink eye, some gastrointestinal issues, all sorts of things. But even here in our electron micrograph, you can see that these are very highly ordered, that they all look relatively the same, and all of the sort of dots you can see on there are the different parts of the capsid coming together to protect the genome. On the right side, we have two examples of complex viral capsids. So this is smallpox up top. Its capsid is this sort of dumbbell or hourglass shaped structure in the middle, right? So it's neither icosahedral nor helical, and it is surrounded on the outside by an envelope. all within these little areas between the capsid and the envelope. There's a whole bunch of proteins in there filling up this space. And down here is rabies virus. All of the members of the same family as rabies have this distinct bullet shape to them. They're actually a helical capsid that's going like this. You can kind of see the lines of it going, woo, it's hard to trace with this little pointer. But they curl up in such a way that they take on this bullet shape, and they do also have an envelope on the outside of them. So rabies, smallpox, Marburg are all enveloped. Tobacco mosaic virus and adenovirus are not enveloped. They are naked capsid viruses. Okay, so there's a lot of variety in genomes and how they can be composed. And how the type of genome they have is one of the main ways that we use to classify viruses. And what most virologists use is this system called the Baltimore classification system. So it was developed by a scientist named David Baltimore. And this system asks a very simple question. How does the virus make messenger RNA? So if you know the genetic makeup of the virus, you know what its genome is comprised of, you can work out how it gets to messenger RNA, and you can put it into its Baltimore class. So I want you to pause the playback really quickly, and I want you to review the steps of central dogma that you learned in genetics. Remember the flow of genetic information throughout the cell. Okay, if you're back with me now, our first group of viruses, the class 1 viruses, are double-stranded DNA viruses. And they follow central dogma just like we do. So this double strand of RNA, it has one strand that's a positive sense that reads just like a messenger RNA, and one that's antisense. So the antisense one gets transcribed into a messenger RNA that's then translated into protein, just like ours. So some examples here would be adenovirus, which we just looked at. Herpes simplex viruses, you've all heard of herpes viruses, and the papillomaviruses, like human papillomavirus. So they work kind of just like us. In fact, these two use our host RNA polymerases to make their messenger RNAs. So in a variation on a theme here, we have single-stranded DNA viruses like parvovirus. They have a positive strand of DNA, and they convert that to double-stranded DNA and then make messenger RNA. So they're going through the same steps that we use, but they have this additional step first. Slightly weirder, but same concept, are our class 7 viruses. I should say virus. There's really only one in this whole group, and that's hepatitis B virus. So the hepatitis B virus does have a mostly double-stranded DNA genome, but it has a big gap here. So we call it a partially double-stranded DNA genome, and bound to it, covalently bound to it, is a small part of RNA. It's one of the weirdest genomes that we have identified to date. But what it does is it starts replicating the DNA here until it converts that gapped double-stranded DNA genome into a regular double-stranded DNA genome, which can then be converted into messenger RNA. Okay? So 1, 2, and 7, those are our DNA viruses. Now, we're going to break central dogma because we need to talk about how those RNA viruses make their messenger RNA. So first up, let's talk about our double-stranded RNA viruses. So these would be things like rotavirus and reovirus, which can cause some gastrointestinal distress. They work similar to how a double-stranded DNA virus works. They have their double-stranded RNA genome, and from that, they have a special polymerase that they code for that's going to make a messenger RNA that gets translated into protein. Then we have our negative strand RNA viruses in class 5. Influenza is the best example of this. So this negative means that it's antisense, and what we need is a positive sense messenger RNA. So this is the opposite of a messenger RNA. So we first need to use a polymerase to convert it to transcribe a positive sense messenger RNA. So the genome is negative. It's used as a template for the transcription of messenger RNAs. Our class 4, we're not going in numerical order here, are our positive strand RNA viruses. So this positive strand RNA gets into the cell and it already looks like a messenger RNA. So it can be directly translated into protein. But we need to make more than just 1 or 2 copies of this so that we have a lot of messenger RNA from which to make a lot of protein. And to do this, our positive sense RNA gets transcribed into a negative sense or antisense RNA that now is going to be made into more messenger RNAs. Okay, so we need a negative sense template to make more positive sense messenger RNAs. So we go from positive to negative back to positive. And there are a lot of positive sense RNA viruses. Poliovirus is one, dengue virus that I work on is another, and of course the coronaviruses, like the one we're dealing with now, are positive sense RNA viruses. Now the last group is the weirdest, and that would be our retroviruses, or our class 6 viruses. These also have a positive sense RNA genome, but they don't make the negative sense RNA. They take their positive sense RNA genome and convert it into a negative sense DNA. That negative sense DNA then is used as the template to make double-stranded DNA, which is then used as the template for the transcription of messenger RNAs. This is the one where you might scratch your head and be like, why did it evolve this pathway? Why didn't it just start with a double-stranded DNA genome or just do this? And that's a really good question. But this is what they do. Positive sense RNA to a negative sense DNA to double-stranded DNA to messenger RNA. Okay. So we've looked at the basic structures of viruses, a genome, a capsid, maybe an envelope. We've looked at the Baltimore classification and how we go to our messenger RNAs. Now let's talk more specifically about the function and the role of the viral envelope. So again, these are lipid bilayers. They are derived from the host. They can be from the cellular membrane. They can be from the endoplasmic reticulum membrane, from the nuclear membrane, and they are embedded with proteins. Now, these proteins that are on their surface are encoded by the virus, okay? So over here we have a coronavirus. If you look in the electron microscopy, you can see just on the outside here, the spikes sticking up. That's actually where it gets its name from. When these electron micrographs were first done, they said it looked like the corona of the sun, the sort of halo that we see around the sun, and they named them the coronaviruses, right? So here's those glycoproteins sticking up on the outside of our membrane. So this is the membrane here in blue, inside of which is the helical capsid surrounding the viral genome, which is the little red line here. Over here we have something like rotavirus, also enveloped, but in this case it has an icosahedral capsid there within the envelope and the genome inside that capsid. You can see this one has a variety of proteins sticking out from it, but all of those are going to be encoded by the virus. And again, viruses that lack an envelope are generally called naked viruses, or they may be referred to as non-enveloped viruses. Now this envelope and the glycoproteins on them are critical for host recognition and attachment. If we get rid of this envelope by, say, putting ethanol on it and drying out those lipids, by using soap and disrupting this membrane, the virus is no longer infectious. So let's take a look at influenza virus. Normally I would stop the class here and ask you to talk to each other and tell me, what do you already know about influenza, or what do you think you know about influenza? And we get a few answers about the type of disease it causes, maybe a little bit about H and N, hemagglutinin and neuraminidase. But here's what it looks like. Influenza virus is an enveloped virus. Sticking out of it are a number of proteins, HA, which stands for hemagglutinin, NA, which stands for neuraminidase. It has a protein, this little blue protein, that links the envelope and the hemagglutinin and neuraminidase proteins together, and inside is the segmented viral genome, which we'll talk more about on the next slide. The interesting thing about this is that the envelope is not really closely associated with these capsids, so the viruses, while we draw them in this nice circle, are actually pleomorphic. They can have a variety of shapes. And you can see that down here in the electron micrograph, where some are like kidney bean shaped, some are oval, some are more actually round. But you can see their HA and NA sticking off of them here too. Right. So influenza is enveloped. It is a negative sense single-stranded RNA virus that has a segmented genome. It is Baltimore class 5, and each of those segmented genome pieces, genome segments, is encased by its own helical capsid that's contained within that envelope. So there are 8 different segments of that negative sense single-stranded RNA. 2 of them get spliced to give rise to about 10 genes. The ones that you have heard of the most are hemagglutinin and neuraminidase, also called HA and NA. And these do, like I mentioned on the last one, they stick out from the viral envelope. And HA is important for attachment, NA is important for release of the virus. Influenza belongs to the larger viral group of the Orthomyxoviridae, and there are 3 distinct serotypes, types A, B, and C. A and B are the ones that we generally deal with every year during flu season. C does not really infect humans. And in addition to HA and NA, there are a few other proteins. NP is the capsid protein. It stands for nucleoprotein. So right here where it says ribonucleoprotein, that means nucleoprotein in complex with the ribonucleic acid. And that's kind of what these little snake-like looking structures that you see in here are. And then there's the M1 protein. The M1 protein, the matrix protein, lies just beneath the envelope and helps to add structure to the envelope and to hold the whole virus together. So influenza gets this really weird naming convention. I don't know who came up with it. It's really clunky and not that great, but it's something like this, where we have an A, which tells us that it's serotype A. Next, we have where that strain was isolated from. So Perth, Australia. 16 is the number of isolates from that particular area. So Perth 16, this was the 16th strain isolated from Perth in the year 2009. And then in parentheses, we give the hemagglutinin and neuraminidase breakdown. So H1N1. So this was an isolate from the 2009 swine flu pandemic. And so for hemagglutinin, there are 18 different types of hemagglutinin, and for neuraminidase, there are 11 different types. So you'll see numbers, or you'll hear numbers like H3N2, H5N1, H1N1, and those are referring to the type of hemagglutinin and the type of neuraminidase in that particular strain of the virus. All right. So once a virus gets into a cell, there can be a number of different outcomes of that infection. So there can be what we call a productive infection or a lytic infection. This is where the virus is going to go through its whole replication cycle that we're going to talk about and wind up making more and releasing more viruses. So prime examples would be influenza and coronaviruses. Viruses can also establish persistent infections within cells. We often call these chronic infections, so an example would be hepatitis B, or latent infections, where we may see a burst of symptoms that then disappear for a long time and then come back later and disappear again. So the best examples of our latent infections are the alpha herpes viruses, such as our herpes simplex viruses, HSV1 and HSV2. And HIV1 and HIV2 can also be characterized as latent infections because they seem to disappear for a long time before they make a reemergence and we see symptomology. Some viruses are also capable of causing transformation of human cells, which is also called oncogenic transformation. So cancer-causing viruses. You are all familiar with human papillomavirus, or HPV, as the most well understood in the general population of viruses that cause cancer. But there are also a number of herpes viruses. They belong to the gamma herpes viruses, like Epstein-Barr, that can cause cancers as well. So there are a number of different viruses that can do this. And then the last outcome is what we call an abortive infection. So this is when a virus gets into what we call a non-permissive cell, a cell that does not support their viral replication, or a non-permissive host. So a host animal that does not support their replication. So maybe this cell is lacking a key enzyme that the virus needs for its replication, and the virus gets in, but then none of the rest of viral replication happens. We call it an abortive infection. Okay. Excuse me. So there are 6 steps to the lytic replication of an animal virus. These are those 6 steps: attachment, penetration, uncoating, biosynthesis, assembly, and release. We're going to walk through these different steps, and at the very end of this slide deck is a diagram that puts them all together. Normally I would draw this on the board for you. I'm going to make one of those videos and upload it here in a day or two. Right. So think about, maybe pause it here and think to yourself, of these 6 steps, which of them is going to be the most important in determining what cell type a virus will infect? So what's going to determine whether your virus infects respiratory cells versus liver cells? Now, if you came up with the answer attachment, you're correct. This attachment phase is what determines whether the rest of these are going to happen. Okay, so attachment requires a couple of things. First, it requires that there be some sort of receptor on the cell surface. So this could be proteins, it could be carbohydrates, it could be lipids. Anything that's expressed on the cell surface can be used by a virus for attachment. So sialic acid, which are these carbohydrate moieties added to a variety of proteins in our lungs, is the receptor for influenza virus. SARS-CoV-2, the current pandemic strain, binds to ACE2, which is angiotensin converting enzyme 2. Now, all of these receptors, even though we refer to them as viral receptors, they have some normal cellular function, right? Angiotensin converting enzyme 2 has a normal function. Viruses have evolved to exploit the fact that these cells are on the surface to allow the virus to attach and get in. Prime example, HIV uses CD4 as its primary receptor. So it can infect cell types that express CD4. So there has to be a receptor on the cell surface, and there has to be some sort of protein on either the capsid or the envelope of the virus that can then attach to whatever that cell surface receptor is. So for influenza virus, it's the protein hemagglutinin that's on the envelope. For other viruses, it may be spikes coming out of their capsid, glycoproteins of various types coming out of their envelopes, or other envelope proteins as well. So here we have influenza virus with its genome segments. And the purple is the hemagglutinin, which attaches to the host sialic acid like this. When that attachment happens, it facilitates entry of the virus into the host cell. So that attachment allows our host cell to facilitate entry of the virus into the body of the cell. Oh, we're missing something there. Attachment of a virus to its host cell facilitates entry. I don't know what happened with that. Okay. And again, that first step of attachment is going to determine what type of cells or even what species a virus can infect. And we call this propensity for a virus to infect one species, one type of cell, one tissue, we call it tropism. So we can have cellular tropism, where a virus can infect one type of cell but not another. So HIV can infect macrophages because they express CD4, but not neurons because they do not express CD4. Influenza has has tissue tropisms. It infects the lungs, and it can infect a variety of cell types in the lungs, but it does not infect the brain. And then we have host tropism, where myxoma virus normally infects rabbits. It's like a papillomavirus for rabbits, but it does not infect humans. Okay, so tropism at the level of what species, what tissue, and even what specific cells can be infected by a particular virus. If your virus can't attach to this type of cell, it cannot set up an infection at all. All right, so attachment is step one. Attachment of a viral protein to a host cell receptor, which allows the virus to get in. And it can get in, this is step two, penetration, through a number of different ways. Those are listed here on the left side of our slide. So viruses can directly penetrate into the cell, and this is generally with non-enveloped viruses. So here we have our virus that's going to attach to receptors, the little red dots on the cell membrane. After it attaches like so, a sort of pore opens that allows the viral genome to move into, be deposited in the cell. The second is that the membranes, so the envelope of the virus and the cytoplasmic membrane of the cell, can fuse. So here we have an enveloped virus, and those glycoproteins on the virus interact with the receptors. This interaction pulls the viral envelope, which remember is just a lipid bilayer like a regular membrane. It pulls it in close proximity to the host membrane. And when two lipid bilayers get forced really, really close together, they will fuse. And so now we have our viral envelope that has fused to our cellular membrane, and the viral capsid, this whole thing, is being released into the cytoplasm of our cell. And then the third method of penetration is endocytosis. So like above, our virus attaches to receptors on the cell surface, but rather than facilitating fusion of the membrane, in this case, our cellular membrane starts to invaginate, and endocytoses are a viral particle. So this picture shows an enveloped virus. A lot of naked or non-enveloped viruses get in by endocytosis also. So once they're endocytosed, now they're in an endosome, and as this endosome matures, the virus will escape, and it can release its capsid from that endosome into the cytoplasm. Okay, so penetration is just how does our viral capsid get into the cell. But if we look here at this middle diagram and we stop right here, we say, okay, well, our viral capsid with the genome inside has been released into the cell. But we still need to get the genome out of the capsid so that it can be used to make messenger RNA, to make protein, and to make more genomes. And that step, the removing of the genome from the capsid, is what we call uncoding, which is step 3 in the viral replication process. So here with direct penetration, as our capsid interacts with the cell membrane, our viral genome gets deposited directly into the cell. So the genome is released immediately. In this one, where our enveloped virus went through membrane fusion, our capsid is released into the cytoplasm. Now that capsid gets removed, gets degraded, and our viral genome is released. And in our third scenario of endocytosis and escape from the endosome, what can happen is that our capsid gets into the cytoplasm and then gets removed, or what also happens is that the capsid will be removed in the lysosome and then the genome is released into the cytoplasm. So a couple of different ways that a viral genome can be released from an endosome or a lysosome. Okay, so steps 2 and 3, uncoding, attachment, penetration, and uncoding. So these two pictures show us a couple of electron micrographs of how viruses can enter into the cell. So here on the left, we have a pig retrovirus, which is being endocytosed by receptor-mediated endocytosis. So this blue in the middle is the DNA, this kind of dark orange is the capsid, and the lighter orange is the envelope. So we can see over here where the host cell membrane in green has attached to, has interacted with those viral proteins, and we can see that this whole thing is invaginating around our retrovirus. On the right side, we have a herpes virus that's entering the cell by fusion of the viral envelope and the plasma membrane. So same color scheme. Blue is the genome, this dark orange is the capsid, and the lighter orange is the envelope. And so you can see, it's kind of harder to see, but the envelope has already started fusing with our plasma membrane, and this capsid and nucleic acid complex is going to be released into the cell. Okay, so influenza virus attaches to sialic acid, triggers receptor-mediated endocytosis, so we can see that here, the invagination of our cell membrane, and now the virus is inside the endosome. Now, if you remember, what happens in an endosome is that endosome acidification is the first thing that begins, and the point of acidification is to denature any proteins in the endosome. So as the proteins on our endosomes start pumping in acid, this does in fact cause a structural change to viral proteins. Okay, so if we look down here, here's our viral HA protein, hemagglutinin, in its normal state. As the internal pH of the endosome starts to decrease as it becomes more acidic, we do see slight changes in the shape of our HA. So these blue parts here are starting to come apart, and these little pieces in the middle are going to undergo a very rapid and very dramatic change as this pH continues to empty. So we get acidification of our endosome, and here within the virus itself is a protein called the M2 ion channel that actually takes this acid now and pumps it into the virus itself. And this triggers our greater change, where these little regions here on the HA are going to flip out and insert themselves into the membrane of the endosome. So these are called the fusion peptides. Okay, so they stick out and they insert themselves into the membrane of the endosome, and then they change even further. So now this variable region here, this kind of unstructured region, acts like a hinge, and you can see in this panel where our fusion peptide and these anchors to the viral membrane are starting to pull together. Now we have our two lipid bilayers in super close proximity. They are going to fuse like this. And now we've created a way for all those viral gene segments to get out of the endosome and be released into the cell. Okay, so here's our fusion event happening. Now our gene segments get out. They actually, for influenza, traffic to the nucleus where viral replication occurs. But we have to have acidification to allow this to happen. So one of the drugs that we use to treat influenza is an M2 ion channel blocker that prevents acidification of the inside of the virus. So none of this ever happens and the segments don't get released. Okay, so attachment, penetration, and uncoating. The next step, biosynthesis, is the biggest, the most complex step. We are not going to worry about this in great detail. We will talk about it a little bit as is necessary. We've already talked about it some when we talked about the Baltimore classification of viruses. So I break step 4 down into a couple of other steps, into mRNA synthesis, which we've already talked about, right? So how do our viruses generate messenger RNA? And this is one of the first things that has to occur. Before you can make more viruses, you need messenger RNAs to make those viral proteins. So typically, the viral genome serves as a template for viral messenger RNA synthesis, right? So for our double-stranded DNA viruses, they are made into a messenger RNA. For other viruses, though, the viral RNA itself is the messenger RNA. So again, with our positive sense single-stranded RNA viruses, like flaviviruses, like dengue virus, and our coronaviruses, those positive sense single-stranded RNAs can act as the first messenger RNA and immediately begin making viral proteins. We'll talk slightly more about that as we talk about our different types of viruses. The second part of biosynthesis is genome replication. This varies greatly by the type of virus. So even though adenoviruses and herpes viruses and smallpox are all double-stranded DNA viruses, they all actually replicate their genome very differently and not necessarily like we replicate our own genome. We're not going to focus on this level of detail in the class simply because it's really not necessary for you guys to know this. And when I teach my undergraduate virology class, we spend about a month just talking about genome replication strategies. So know that they do replicate their genome because they need more genomes to put in more capsids to make more virus, but we're not really worried about the how, okay? And then the final part of biosynthesis is protein synthesis. We can break protein synthesis up into a few different times, I suppose is the way to say it, during infection. So some people break it up into immediate early, and late protein synthesis. And this can all be delineated by whether or not genome replication has happened. So those immediate, and early proteins that are made are usually enzymes. And these are either enzymes that are needed to replicate the viral genome, like a polymerase that's needed to make more viral genome, or they're enzymes that are going to modify the cellular environment. So they're going to push the cell into S phase so that the cell is making more nucleic acids of their own so that we can take, or viruses can take those bases away. Or that they're going to turn on protein synthesis so we can make a lot of viral proteins. Genome replication, when we switch from these early proteins to making genomes, that sort of delineates the demarcation between early and late protein synthesis. And the late proteins are what we call the structural proteins. So the proteins that make up the capsid, any envelope spike proteins, matrix proteins, those things that make up the structure of the virus itself. And we make these in huge amounts so that we can make a whole lot of viruses. I keep saying we. I'm not a virus. Viruses do this, not me. Okay, so early proteins are enzymes, going to modify the cellular environment, going to be used to replicate the viral genome, and late proteins are the proteins that make up the virus itself. So let's look at and put all these together and look at how influenza does this. So up here is step 1. We have attachment of our influenza virus to our host cell. So like this would be our HA, it attaches to sialic acid, and that triggers endocytosis. So we've got attachment, we've got penetration, and now we're going to have uncoating, because as this endosome acidifies, our gene segments get released, and they're going to travel for influenza into the nucleus of our cell. Here in the nucleus is where all of our biosynthesis is going to take place. Okay, so we have mRNA synthesis, we have viral genome replication here, and we have splicing, because a few of these messenger RNAs do get spliced. Like with any other messenger RNA, these are going to get shipped out to the ribosomes and to the endoplasmic reticulum for translation. Some of these, the glycoproteins, hemagglutinin and neuraminidase, along with matrix proteins, are going to then travel through the ER and the Golgi and be put on the surface of the cell. All right, so they are here, they are associated with the cell's plasma membrane. Some other proteins, like polymerases, like our nucleoprotein, the capsid protein, go back into the nucleus because this is where new genome segments are made. So for flu, these genome segments and the capsid and everything gets assembled here in the nucleus and then shipped back out to the cytoplasmic membrane. And here, all of our gene segments are going to interact with those proteins, those viral proteins on the cell membrane. They're going to interact with the matrix proteins that we can see right here. This is what we call the step of assembly. And then the final step is release, where this virion now leaves the cell. And influenza does this by a process called budding, where it's essentially the opposite of endocytosis. So here we have invagination to take it in during attachment and penetration. And here we have sort of the extrusion of the cell membrane around the virus. So HA and NA, hemagglutinin and neuraminidase, are the proteins on the surface of our influenza virion. And this is the place where neuraminidase is important. Okay, so think about it. HA attaches to sialic acid on the cell. So if you have all these HA molecules right here next to sialic acid, our influenza viruses are just going to stick to the cell they're coming out of, like Velcro. They're not going to just pop off quite so easily. They're going to stick here. Neuraminidase cleaves, it cuts up sialic acid so that now the virus can get off the cell and go on and infect a new one. So some of our other drugs that treat influenza inhibit the activity of neuraminidase so that we kind of get stuck at this part and we never get full release of the virus into the environment. Okay? All right. So the last two steps, assembly and release. DNA viruses, for the most part, assemble in the cytosol and then are released from the cell. They can also assemble in the nucleus, and then they must be released from the nucleus into the cytosol and then eventually get out of the cell. It just depends on where they carry out biosynthesis. Do they do that in the cytoplasm, like smallpox? Do they do that in the nucleus, like adenovirus? Most RNA viruses assemble in the cytosol because that's where most of the replication is. Flu is kind of a weird one that it replicates in the nucleus. Most of them replicate just in the cytoplasm or associated with the endoplasmic reticulum. So influenza is an exception. And there are three methods for release. The first is that they can just lyse the cell, like when we talked about bacteriophages, and they just cause the cell to burst open, like this, and now you have a whole bunch of new viruses floating around in the environment. So this is what our naked viruses may do because they don't need an envelope. So you just burst the cell and you get out. So adenovirus does that. It makes a protein called the adenovirus death protein that causes lysis of the cell. The second method is exocytosis. So this can be for naked or enveloped viruses where they are exported through the ER and the Golgi and released into the extracellular space. If a enveloped virus leaves by exocytosis, it's going to get its envelope from the endoplasmic reticulum. So dengue is a good example here. It gets its envelope from the ER and leaves by exocytosis. And then finally, we have our last one, like influenza, where we can have budding from the plasma membrane, where the virus, the capsid associates with the membrane and it extrudes and buds out. Regardless of which way a virus gets out of the cell, eventually the cell is going to die because you're using up all its resources. These will die really quickly because you're bursting your cell wide open. These might be a slightly slower death because death is due to loss of resources and loss of ATP rather than bursting of the cell. So these images just kind of give us an idea, show us a bit more clearly how budding works. So just real quickly, here we have a viral capsid that associates where the viral glycoproteins are, usually on some sort of raft in the membrane. Our membrane then begins to extrude, continues to do so until finally the whole virus is released and is free in the extracellular space. And down here is an electron micrograph of HIV-1 that shows this really nicely. Here we have our viral proteins in orange. They've associated with our viral capsid in green, and it continues to pinch away from the cell, which is the kind of pink and yellow thing, until the whole virus has been released. All right, so this is another good place to pause and to think through these questions on your own. So how is the cell going to be affected by viral replication other than just budding, right? So what processes, what cellular processes are going to be altered or impaired during viral replication, and what's going to happen to the cell, right? I kind of already said cells are going to die eventually because of loss of resources, loss of bases, loss of amino acids. But what about the immune response? How might that play a role in viral replication or pathogenesis? And then what happens to tissues and organs or organ systems during an acute viral infection? So pause here, think about these questions for a few minutes. Now let's go back to the case study that we started this with, right? That was a long time ago now, so let's think back. There was a local nursing home experiencing an outbreak of bacterial pneumonia in its residents. And in the previous 6 weeks, there had been a similar outbreak of a viral respiratory infection in the facility. And I asked you what you thought was causing it and why. Well, here's your answer. The primary infection was influenza virus. Why were the people predisposed to secondary bacterial pneumonia? Tissue destruction and cell death are the results of viral infection, viral replication, and also the result of the immune response to viral infection. So during the acute replication and infection of the virus, those lung cells are being destroyed by the virus and also being killed off by the immune response. So during the convalescent phase of influenza, which can last for about 3 months, patients are more susceptible to further lung infections, and in particular to bacterial lung infections. So let's look at why that is the case. Here are 2 pictures of normal respiratory epithelial cells before and after influenza infection. So remember, We have these ciliated cells, ciliated epithelial cells in our lungs that help clear mucus. Influenza kills those cells off. So here's all their cilia on the left, and over here on the right, those cells are dead. Our epithelium is denuded. You can't effectively clear mucus. You can't effectively clear pathogens. And these cells can take months to regrow. So for about 3 months post-influenza infection, a patient is incredibly more susceptible to secondary bacterial pneumonia. Okay. So you guys have all learned a little bit about influenza, and you've heard that usually it affects older people and young children. That's why normal, healthy individuals like us should be getting our flu shot to protect those who are unable to fight off influenza. What the virus does is, again, it infects the respiratory ciliated cells. And one of the ways that it leads to cell death is that it shuts down host protein synthesis. So it does this by a process that we call cap snatching. So the virus steals the 5' methylguanine caps from host messenger RNAs and sticks it on its own messenger RNAs. So only viral messenger RNAs have a cap. Only viral messenger RNAs get translated. Host proteins don't get translated at all. It's pretty cool. They also cause the release of lysosomal components. And this shutdown of host protein synthesis, the release of lysosomal components, leads to apoptosis of the infected cells eventually, which again leads to local inflammation there in the cell. So here we've got some cells that are infected. They're dying. They're causing an immune response of various types. Now, while the death of those cells causes some damage to the lungs, the majority, and this is true for a lot of virus, the majority of pathogenesis is actually host-mediated. So it's caused by your immune response. With influenza, back, if you've heard anything about the bird flu, that was H5N1 back in the 1995 or so, this strain of influenza was really, really weird. It caused what we call a cytokine storm. And that's actually what caused major damage. We saw a similar effect with the 1918 pandemic, the Spanish influenza pandemic, as it's been called, where the immune response was so strong, and that's mostly what led to death. So as the virus is replicating, we get pro-inflammatory cytokines. They're in the lungs. We have macrophages that are coming in to clear up our cells, the activation of T cells, and the production of many cytokines. You guys have learned all of this sort of stuff. And the cytokine levels were so high that it led to immense tissue damage in the lungs, necrosis of lung tissue, tissue destruction, influx of our white blood cells, and dilation of blood vessels. And so the deaths that we saw with the H5N1 bird flu and in the 1918 pandemic happened particularly in young, healthy people with robust immune responses, because all of this damage was immune-mediated. People who were older or younger, or whose immune system didn't work as well, they were not really affected by either of these two strains of flu as much as young, healthy people. And then, of course, even with our seasonal influenza, one of the big fears is death. Usually, death due to seasonal influenza is when there's some underlying condition, immune disorders, heart problems, pre-existing respiratory distress, or superinfection, meaning, like in the case of pneumonia, a person has influenza concurrent with or followed immediately by some sort of other infection. So we call that superinfection. Okay, this is a long one. Sorry, guys, bear with me. The symptoms of influenza we are all familiar with. You've heard them. We call them flu-like symptoms. They begin with fever, general feeling bad, usually a headache, and then those muscle aches that are really symptomatic of influenza. Usually coughing and sneezing, often a productive cough where you're coughing up phlegm and you're sneezing out a lot of mucus. And flu can usually become accompanied by diarrhea as well. It's estimated that each year in the United States, there is a loss of 71 to 167 billion dollars in lost wages due to people missing work for influenza, and anywhere between 5 and 10% of the population gets infected with flu. So this little triangle from the CDC here is showing the data from the 2017-2018 flu season, which was a particularly bad one. But you can see the estimated deaths, and these numbers are still being crunched right now. The estimates range anywhere from 12,000, which is pretty low, to 61,000. It's believed that they're probably toward the higher end of that range. We had a lot of hospitalizations due to flu and flu confounding problems, and a whole lot of flu-like illnesses where people go to the doctor, they get tested, they test positive for flu, and they go home. They don't need hospitalization. So it's a really big burden on our healthcare system every year from October to about April. In the 1918 pandemic, the Spanish influenza pandemic, 50 million people died from the flu in an 18-month period. That's more deaths than World War I and World War II combined. Fortunately, so in 1918, we didn't really have anything to treat it. We weren't doing quite as well on that sort of stuff. Now we're much better at diagnosing and treating. As you all know, there is a rapid flu test. This only works if you're in the acute phase of infection. You need to have a lot of flu there for the test to pick it up and test positive. And as you guys may know, these actually test negative fairly often, so they're not that reliable. But there are a number of treatment options now. Supportive treatment is usually what people are told to do. Go home, take some ibuprofen for your fevers and chills, drink lots of fluids, all those good things that you should be doing when you're sick. And we have a couple of other drugs that we use. So oseltamivir, or Tamiflu, as its over-the-counter name, is a neuraminidase inhibitor, like we already talked about. There's others that you may have heard of, amantadine, and rimantadine. So these target that M2 ion channel. They only work against influenza A. And then in addition to Tamiflu, there's also Relenza. Its sciencey name is zanamivir, which is also a neuraminidase inhibitor. The big issues with these drugs, though, is that you really need to start taking them within about 24 hours of the onset of flu symptoms for them to do much to shorten the duration of disease. If you go into your doctor after you've been sick for like 3 days and you start taking them, you're already on the downward slope and they don't do much. So you need to start taking them really, really soon after you start showing symptoms. So, pros and cons. And of course, we can prevent influenza pretty well with our annual vaccine. So the vaccine is made of whole killed virus. It is raised in eggs, so there are a couple of groups of people who should not take the vaccine. That's anyone who's allergic to eggs and anyone who has experienced Guillain-Barré syndrome in response to a previous flu shot or a previous vaccine that's been raised in eggs. So it's whole killed virus. Most of the immune response that you get is to the hemagglutinin and neuraminidase proteins. There's been pushed for years to develop a universal vaccine that will work against all strains of influenza, H1 through 18, N1 through 11. And most of these use the M1, the matrix protein, as their target. So that protein that keeps the envelope associated with the virus. Back when I was a postdoc at Tulane, we worked a lot on testing many of these vaccines in ferrets to see if they would protect against the flu. None of them did. M1 is not, I don't know, maybe one day it'll work. I think the M1 is just not an immunogenic enough antigen, and so we're having a hard time making it work. The other big issue with the flu vaccine, as you know, is that we don't really do a good job of getting people to get their vaccine here in the US. So this is showing us over time the coverage of vaccination. So this like NIS and BRFSS, those are surveys that were done where people were called and asked whether they got the flu vaccine for their children or for themselves. And so what you can see if we look at our children, which are our dark lines, Americans are doing a decent job of getting their kids vaccinated for flu every year, right? Because we're always told that flu can be deadly in young children. So if we look in 2011, 2012, it was 50%. For the 2018, 2019, it was 62%. But we're not doing as good at getting adults vaccinated. So these are our bottom two lines. I mean, look at this. In 2011, 2012, not even half the population was getting their flu vaccine. And even in 2018, 2019, only between 45 and 50% of respondents said they got the vaccine for themselves. Interestingly enough, we see this sort of bump for 18-19. The 2017-2018 flu season was one of the worst that we've seen in a while. There was something like 61,000 deaths due to influenza that year. So it seemed like people took it more seriously the following year. And we just need to get these numbers to keep going up. Okay, last couple of things. Whew, this is a long one. Genetic drift and genetic shift. Genetic drift is something that all organisms can do. Genetic drift is just random mutation that allows for genetic changes. In viruses, viral polymerases tend to be of lower fidelity, meaning they're not as good as ours. They make more mistakes than ours, and particularly RNA viruses, because viral RNA polymerases that replicate those genomes, they don't proofread. So if they make an error, the error stays. So genetic drift is where we have one type of virus, some mutations occur, and we still have that same species of virus now, but there's been some change to it, like maybe it makes it slightly more transmissible. So that we can see this in all viruses. And particularly, this is what allows for evolution in our non-segmented viruses. So HIV is the prime example. Its polymerases are really terrible. They make so many mistakes. It mutates really, really quickly. Herpes viruses, which are DNA viruses, they mutate much more slowly because DNA polymerases do proofread. But also rabies, which is a negative strand RNA virus, and its RNA polymerase is pretty faulty as well. So genetic drift, that's just mutation over time. It takes a very, very long time for enough mutations to occur to cause a big change in the viral phenotype. Genetic shift, on the other hand, is a combination or is a consequence of having a segmented genome. Okay? So another word that we use for genetic shift is reassortment, because we are reassorting the genomes between 2 or 3 different species of the same virus. Okay, so here we have a human and a duck. So our human here gets infected with the human influenza virus, which is the type H2N2. And our little ducky here gets infected with the avian influenza virus, which is type H3N8. Now, the bird flu cannot directly infect humans, and the human flu cannot directly infect birds because the sialic acid in our lungs is different. So we have sialic acid moieties that human flu binds to. Ducks have and birds have sialic acid moieties that bird flu binds to. Bird flu can't bind to ours, human flu can't bind to theirs. But pigs have both these types of sialic acid. So human influenza can infect a pig, bird influenza can infect a pig, and there in the lungs of the pig, these two different types of viruses can swap out genome segments. So we get a unique gene combinations from more than one strain of virus. Okay? So we get a new virus that now is human H3N2. Now this can infect humans, and it can go on to cause a new disease Maybe it causes a pandemic strain like the H1N1, 2009 swine flu influenza, or the H5N1 bird flu, right? So that's how these arise. We need this unique setup where we have humans, birds, and pigs living in close contact so that the pig can act as a mixing vessel to give us a new recombinant strain of the virus. And this genetic shift, it's a very fast mutation, right? We don't have to wait for the accumulation of small mutations. It happens really quickly. So we only see this with our segmented viruses. So our influenza with their 8 segments, hantaviruses with their 3 segments, and reoviruses with their, I think they have 10 different gene segments, can all undergo genetic shift and give rise to a new strain really, really quickly. So if you had asked anyone, a year ago, what would be the next human pandemic, they would have said a reassortment influenza virus. And yet here we are staring down at a pandemic caused by a virus that can only undergo genetic drift because it's not segmented. This diagram is just to kind of give you another view of genetic drift at the cellular level. So here we have like the L strain of influenza and the M strain of influenza. Both of these have to infect the same cell at the same time, not just the same animal, the same cell. And now within this cell, we have replication of both these types of viruses and all their different genome segments. Now, when the virus assembles and buds, it doesn't really care where these different segments come from, what virus they originally came from. All it cares about, to anthropomorphize a virus, is that it gets all 8 segments. So a lot of times you'll get things looking fine. We'll get a lot more L virus out, we'll get a lot more M virus out. But here in this one, we've swapped out one segment. And that's usually all it is one segment gets changed and it gives rise to a new form of virus. So this one is called the R3 for whatever reason. So we've swapped out gene segment 2, and all the rest of them remain from the M virus. And this gives us a new strain. Okay, this is the last slide. Again, this one, it just kind of is to put together our discrete phases of viral replication, from attachment and penetration to uncoating of the viral genome, to replication, where we make more genomes, transcription, translation, assembly of our genomes with our capsids, and release, in this case, by budding. This, again, is what we call the lytic cycle of replication because it results in cell death. Okay, that's it for this one, guys. Next one I put up will cover pathogenesis and antivirals.